to talk about a problem that can occur if it's really hot and dry. And this is mostly happening in plants that we are calling C3 plants. So I forgot to mention in the last video why things were called C3 plants, and I'll mention that in just a second. But anyway, the problem is happening when it's hot and dry, and what will happen is the stomata will close. And when that happens, there's no carbon dioxide source. So let's talk about how this works. But first, I'll just kind of go back over this cycle a little bit, just to talk about the C3. The reason why this, before the pink is added in here, it was called the C3 cycle is because the first stable compound is a 3-carbon molecule. And you're going to see that the C4 cycle that we are going to talk about next, the first stable compound is a 4-carbon molecule, so that is called C4 plants. Okay, so anyway, this has to do with this process, or this thing that we're talking about for photorespiration. Photorespiration, like I said, happens when the stomata close. And what will then go down, instead of carbon dioxide being brought in, instead of that, oxygen will bind at that binding site, and no carbon will be added to this molecule. So we'll get a 5-carbon molecule here instead of a 6-carbon molecule. All right, so this is happening because the oxygen is going up. That is, remember, the light reactions are generating oxygen. However, in this case, because the carbon dioxide is cut off, then we don't have a carbon source for this part of the cycle, this Calvin-Benson cycle, because the stomata are closed, so we only have the O2 then joining in. Now, Rubisco has an affinity for both of those things, so this is why that happens. And you'll get a 5-carbon molecule, and when it breaks it into pieces, one 3-carbon piece will go this way, but the other 2-carbon piece is going to go and be respired. And this is why this process is called photorespiration, because this respiration then is a loss of carbon from the system. Now, this respiration or photorespiration process happens in an organelle called the peroxisome. So I don't really need you to memorize any of this, but basically what's happening is these 2-carbon pieces are going to go to the peroxisome, also called a microbody, and that peroxisome will then break it into, or change it slightly so that it can then enter the mitochondria and be respired. So that's what that's about. Okay, to counteract that problem, we have something called C4 plants. So we've been talking about plants that have an internal structure, with the mesophyll being palisade parenchyma, and the spongy, the veins, like we talked about before, and the carbon entering in through the stomata. Now, in order to counteract this process, what I have to do is I have to remove this accumulation of O2 in here as the stomata close from the photosynthetic cells. In other words, from the cells that might be doing the carbon fixation or Calvin cycle part of the cycle. So what happens in C4 plants, or plants that do that, is they have 2 layers of cells that are doing photosynthesis, where in the outer layer, the oxygen will not interfere with the uptake of any carbon dioxide that's present, and the inner layer is where the Calvin cycle is occurring. So this is found in what we call monocots, or C4 plants. These are things like grasses and palms and things like that. Okay, so what literally happens, so here's that configuration again, the 2 layers, and the 2 layers, the outer layer is called the mesophyll cell, and the inner layer is called the bundle sheath cell around the bundles of veins. Okay, so the vascular system is the purple, bundle sheath cells are the darker green, and the mesophyll cells are towards the outside. So this is the part that's exposed to carbon dioxide and oxygen. Now, the difference here is that PEP carboxylase, which is the enzyme that catalyzes this reaction, does not react with oxygen. All All right, so it is, even though there's a lot of oxygen perhaps present because the stomata are closed, it won't take up oxygen. It only has an affinity for carbon dioxide. So any carbon dioxide that's present can then be turned into a 4-carbon compound called oxaloacetate, or oxaloacetic acid, same thing, and then converted into another 4-carbon compound called malate. And then that can enter the bundle sheath cells via the plasmodesmata and do the regular Calvin cycle. When that 4-carbon molecule donates its carbon to the Calvin cycle, it will donate 1 carbon at a time, leaving a 3-carbon molecule, pyruvate, that will then go back and be regenerated as PEP, which can then take up the carbon dioxide and become oxaloacetate. All right, so this is a cycle that does not have any kind of affinity for oxygen at all. And because the oxygen is out here in the leaf space, it will not interfere then with the Calvin cycle that is inside, and photorespiration will not happen or be completely reduced. All right, so this is what we call C4 anatomy, because the first stable compound is a 4-carbon molecule. Okay, so then if you were to compare C3 and C4 plants, corn is a typical C4 plant, and beans are typical C3 plants. If you compared them over the day on their carbon dioxide uptake, the bean plant does really well, but corn is going to do slightly better. Okay. Now, another thing that can happen is, for a similar reason, for the photorespiration reason, is something that we call CAM plants. So you could say that the C4 plants, what we were just talking about, reduce photorespiration by doing a spatial separation of the steps, okay? But in something that is a CAM plant, instead of doing it in physical location, it's going to use time as its separation. So what happens in CAM plants is they only have their stomata open at night. So at nighttime, it takes in the carbon dioxide and converts it to an organic acid. And then that organic acid during the day, this is basically like a storage molecule. And then during the day, when the sun is out and the Calvin cycle has enough energy to run, it can use the carbon from this organic acid to run the Calvin cycle. So this one is a temporal separation of the steps.